Yes, everybody. I guess we better do something. So today is the 13th. Oh, we're way behind schedule, aren't we? Supposed to be doing chapter 6 and 7. We haven't even started chapter 4, have we? So that's okay. We can just skip some chapters or something. Catch up. Let's do every other chapter. Well, we had an exam last week. I got the exams graded to give back. If you want them, we can do. You can pick them up at break time or something. Or if you want to, we don't have a lot of extra time, but if you wanted to go over what the answers were, guess we could try to do that. But and homework is due today, so you turn that in. And I guess we need to finish up chapter 3 still, right? Anybody remember where we left off? What we were talking about?
I swear it was like the second to last slide.
Oh, okay, good. Oh yeah, I remember. Yeah, so I guess we talked about endocytosis and exocytosis. So these are called bulk movement because instead of just one atom or molecule at a time coming in or out, this is a whole particle, solid particle, or a droplet of liquid being taken in by the cell. So the cell changing shape and so that it reaches around and surrounds and engulfs the particle and takes it in, and now it's in a vacuole or a vesicle inside the cell. If it's a solid particle, like a bacteria cell or something, that would be called phagocytosis. If it's a droplet of liquid, it's called chemocytosis. Amoebas are really good at this. The amoebas, the whole organism is constantly changing shape, and so that's how they eat. They just surround the particle and take it in. Some of our white blood cells can do this, of our immune system. And exocytosis is the opposite. If a cell produces something, it'll put it into a vesicle, and then that vesicle will move to the surface of the cell and it just spits it out. So this is what in humans cells of our glands. A gland is an organ that produces something that is then released from the gland into the body or onto the surface of the body or something. So cells in salivary glands would make the saliva into it, put it in the vesicles, and then release it into the oral cavity, the inside of the mouth. Same thing with sweat glands, earwax glands, hormone glands. This is also called secretion, when a gland cell makes something and releases it by exocytosis. Okay, this concentration gradient, like we learned that diffusion happens because of a concentration gradient, a difference in concentration of something on one side of a membrane or in one place compared to another place. the stuff likes to spread itself out, and it moves from where it is in high concentration to where it is in low concentration. So we would say that's moving down the concentration gradient. And the difference in concentration can be just in the number of atoms or molecules. There's more in one place compared to another, or it could be the electrical charges if they are ions. So if there's more positive ions on the right side than on the left side, and more negative ions on the left side and on the right side, we'd say there's an electrical gradient. More positive charges on one side of the membrane than the other. So those two combine, the combination of the chemical gradient, the concentration of the number of atoms or molecules, along with the difference in electrical charges. That's called the electrochemical gradient. And this is what drives diffusion. Okay, any questions about that chapter? So this was kind of an introductory chapter on cells. We're not done with cells. We're gonna some of the future chapters are about functions of cells, how cells divide. The what? Yeah, the homeworks. Yeah, just start a stack here. So I hope that was interesting at some level. So I think it's an important story. Everyone should know about that. Fortunately, we don't always learn from past stories like that. Okay, so we need to move on to chapter 4 and 5, which are together on one PowerPoint. Recording this, and I wanted to make sure that I started the recording. Okay, these two chapters are together on one PowerPoint because they're very similar. uh, the processes discussed are very similar, except they're opposites, as we'll see. Um, Photosynthesis is just about the direct opposite of aerobic cellular respiration, which is described in chapter 4. Okay, I don't know who this person is, who um, this quote is attributed to her. So it's very true that what you eat can either make you healthy or it can be slowly poisoning you. Okay, bioenergetics. Bio means life, and so this is the study of how energy flows through living systems such as cells, but also this can involve how energy flows through whole ecosystems. Um, so energy comes in from the sun, it's absorbed by plants, and they use it to make organic compounds, and then and that energy from the sun is stored in those compounds that the plant makes. And then that energy is passed up through the food web as the plants are eaten by animals, and then they're eaten by other animals, and so on. This term metabolism, I think we've learned that already. All the chemical reactions in an organism, and that includes synthesis reactions. Of course, synthesis in chemistry means making molecules. And in biology, we call that anabolic. So chemical reactions within your body that are making molecules, we call them anabolic reactions, or anabolism. In chemistry, decomposition means breaking molecules apart, and in biology, we call that catabolic. So catabolic reactions or catabolism, that's what's going on in your digestive system right now, breaking down food molecules into smaller ones. This term metabolic pathway just means one chemical reaction after another, and the products of one chemical reaction are used as reactants in the next chemical reaction. And lots of these metabolic pathways going on in our bodies. And most of these important chemical reactions require enzymes. We'll talk about enzymes a little more in this chapter. Many genetic disorders that can be really devastating, that can even be fatal. In some cases, a baby will not even make it till birth, or will be born with one of these genetic disorders that will lead to a lot of medical issues, or won't live very long. That these kind of genetic disorders I'm talking about involve genes for enzymes. Most genes have the information for making a type of protein. And if you have a genetic mutation in a gene for making an enzyme, which is for an important chemical reaction in one of these metabolic pathways, now your body cannot make that enzyme. So the metabolic pathway is trying to happen, so chemical reactions producing a product, and then that's used in the next reaction, and then products of that are used in the next reaction, but now it comes to a reaction that can't happen because you don't have the enzyme for that reaction. That can have severe consequences in your physiology. And some of these, there's one called PKU, phenylketonuria, which is one of the more common genetic disorders. People with that are missing an enzyme, so so an amino acid builds up in their bodies. It doesn't get broken down like it's supposed to, and that can cause severe problems with their nervous system development. They can be mentally disabled and die young. from that. Okay, anabolic reactions usually require energy. So putting atoms or small molecules together to make a bigger molecule usually requires energy to do that. And then the energy will be stored in the bonds holding that molecule together. Catabolic reactions usually release energy when the molecules are broken apart. Here's what I was mentioned about energy flowing through an ecosystem or a community of organisms. So energy comes in from the sun. It's absorbed by plants, use it for photosynthesis to make organic compounds, sugars and starches and cellulose and proteins and fats. And those molecules become the tissues of the plant. And also, the plant uses some of those molecules for its own food. But there are photosynthetic organisms we call producers, so they produce their own food. Consumers, that's us, and organisms that can't do photosynthesis, and they have to consume other organisms, other plants or animals or fungi. So when we eat stuff, some of the chemical energy that was in the plants is now in the animals. Everything dies eventually, and everything, well, animals make poop, and so the decomposers eat the dead organisms and the poop that comes out of animals. Decomposers they're also consumers, but they eat dead stuff. They break down dead organisms. So that includes fungi, many bacteria and worms, and some insects. And so this returns the atoms and molecules to the soil, which is taken up by the next generation of plants. Start this all over, but so we say that the plants use energy, being alive, animals like us, we use energy, but energy doesn't disappear as we use it. It's just converted into a different kind of energy. So, like when I flex a muscle, every time I contract a muscle, that generates heat. And so what happens to that heat? It's just lost to the environment. So all of this energy that we organisms are using, it eventually ends up, or almost all of it ends up as heat in the environment, just spread out. So the energy comes in from the sun, it passes through all these organisms, and each organism is using that energy, and that energy just ends up spread out as heat in the environment. Okay, so what is energy? It's kind of hard to define, but the ability to cause some kind of change. So the energy is the described as a capacity to do work. Work means moving a force through a distance, or raising a temperature, producing light. But these are all types of energy. An object that's moving has energy, and anything that has any heat in it that's heat. That's a type of energy. Light is a type of energy. Electricity. Thermodynamics is the study of energy. When I was in college, I had to take a whole year of thermodynamics because I was a mechanical engineering major. It was required. So a couple of fundamental laws of thermodynamics. Energy can be converted from one form to another, but cannot be created or destroyed. I kind of mentioned that a minute ago. Energy doesn't get used up. It doesn't disappear. It's just converted from one type to another. So that's what an electric light bulb does. It converts electrical energy into light energy. Electric motor converts electric energy into energy of motion. Um, Second law of thermodynamics. Whenever energy converts from one form to another, some of the energy is lost as heat. That means that, for example, converting energy from electric to light is never 100% efficient. Like a light bulb, especially the old style incandescent light bulbs, they get hot. That heat is just wasted energy, because what we want is all of that electricity flowing through that light bulb to be converted into light. But any heat that's generated, that's wasted. That's just wasted energy. The fluorescent bulbs, like in here, and neon, or neon, but LEDs are more efficient. They don't get hot. So more of the electricity is converted into light and less is wasted as heat. Electric motors are not 100% efficient, but they get hot. That heat is wasted. We would like all the electricity to be converted into the motion of the spinning shaft of the motor, but some is lost as heat. Your car engines are very inefficient. The gasoline engine, even though that design has been around of the internal combustion engine has been around for way over 100 years, it's gotten more efficient. We got now computer-controlled electronic fuel injection that is way more efficient than the old style carburetors that would just squirt gasoline into the engine. But still, just think of all that heat produced by a car engine. That's all wasted energy, inefficiency. All the heat coming out the tailpipe, all the engine produces so much heat that it has to have its own cooling system with coolant, water and antifreeze mixed together, flowing through the engine to take the heat to the radiator, where there's a motor or there's a fan blowing air through the radiator to get rid of that heat, which is just wasted energy from the engine. And the engine has to have its own water pump. Some of the energy from the engine is used to run the water pump to pump the coolant, and energy is used to spin that fan. So even more, you're wasting more energy to get rid of the wasted energy of the heat. So gasoline car engine, it's only maybe 40% efficient. Okay, so energy types of energy can be put into these two categories: kinetic and potential. Kinetic. The word kinetic means motion or moving. So kinetic energy is energy of motion. So anything moving has kinetic energy. Even heat energy is a type of kinetic energy, because it's the energy of the motion of atoms and molecules just moving around. The faster the atoms and molecules are moving, the more heat energy there is, the higher the temperature. So anything that's falling has kinetic energy. Potential energy is energy that's not doing anything right now, but it could cause something to happen. Like energy stored in a battery, it's not doing anything right now, but if you hook wires and a light bulb up to it, that energy could be converted into light. Hook it up to a motor, it could be converted into kinetic energy. What type of potential energy called gravitational potential energy is what we could say that I'm holding this remote 3 feet off the ground, off the floor. So we could say it has potential energy in it right now, just by being 3 feet off the floor. Because all I have to do is let go of it, and it would start falling, and that potential energy would be converted into kinetic energy. The lower it gets, the closer to the floor, the less potential energy it has, but it's accelerating, moving faster. So it's farther it falls, the more kinetic energy it has. Water behind a dam is a type of potential energy because gravity could make it fall if that dam were not there. Compressed spring, old style clocks that you wind up, that's storing energy in that spring. And the chemical energy in molecules is a type of potential energy. And that's especially important in what we're going to cover later in this chapter. Okay, more about enzymes. Enzymes are very important. Enzymology is a whole science in itself. Some people spend their whole careers just studying enzymes. But remember, an enzyme is a special kind of protein molecule that causes chemical reactions to happen. And there's lots of different enzymes in your body, and each type of enzyme works for just one kind of chemical reaction. And also recall that protein molecules, every protein molecule has a specific shape, a three-dimensional shape called the conformation. And in the case of an enzyme, that the shape of the molecule includes this keyhole called an active site. And this keyhole is a specific shape that the reactants molecules can fit into that keyhole. And in the case of chemical reactions that are done or catalyzed, or they happen because of enzymes, the reactants are called substrates. So this would be a molecule. So this could be like a double sugar, like sucrose. It's made up of two simple sugars together. But that molecule of sucrose fits into that active site perfectly. And so this enzyme, its job would be to break this sucrose molecule in half. So the sucrose molecule would temporarily bind to the enzyme, and then the enzyme would break the bond between the two simple sugars, and then release them, release the two smaller molecules. And now the enzyme is not used up in this reaction. It's ready to grab another substrate molecule, break it in half, and then another one, and then another one. It can do this hundreds or thousands of times per second. So these are very, very efficient molecules. And so a lot of chemical reactions in our bodies would just not happen without enzymes. Remember what we learned, that protein molecules have that special shape called their conformation. But every protein molecule, including enzymes, have a favorite set of conditions that they like to be in. They like to be in certain temperature, certain pH, certain salinity or saltiness of the solution. And it's different for different proteins. Some proteins like to be in acid, acidic solutions. Some like to be in high temperature, some like to be in low temperature. But the ones in your body, they like to be at about 98 degrees Fahrenheit. That's your body temperature. The pH, we learned about pH, and why we learned about pH in a biology class is that the pH of a solution is very important. It affects the chemical reactions going on in that solution. So it's important that the pH in your body fluid stay about 7.4, because if it gets much below or much above that, the enzymes in your body will start to unfold and lose their shape. We call that denaturing, when a protein loses its shape. It's denatured. So If your body's temperature starts to drop, or the pH in your body fluid starts to get too high or too low, your enzymes start to unfold. All these chemical reactions caused by the enzymes will slow down or stop, and that's not good. Cofactors and coenzymes. These are just helpers for enzymes. The difference is cofactors are just atoms, just single atom, like an iron atom or ions, an iron ion or a zinc ion. But coenzymes are molecules, organic molecules, and that's what many vitamins are. They're cofactors or coenzymes. Why do we need vitamins and minerals in our diet? Why do some people take nutritional supplements, take vitamin and mineral pill every day? Many of those are required because they are important cofactors and coenzymes for enzymes in our bodies. And we don't need a lot of zinc or a lot of vitamin E in our bodies, but if we don't have any, if we have a deficiency, then some of our enzymes won't work. Chemical reactions won't happen that are supposed to. Okay, the rest of this chapter is about ATP. I mentioned this in the chemistry chapter. Adenosine triphosphate, these are like tiny rechargeable batteries in cells. Every cell of every kind of organism we know of uses ATP for storing energy temporarily in cells. And the rest of this chapter is how does that happen? How does a cell recharge these little batteries? And that's, well, let's look at ATP, the molecule, and it's called adenosine triphosphate. This is the adenosine part here, and then there's 3 phosphates here. Why it's called triphosphate. Tri means 3. And this third phosphate is held on by a bond and that's the stored energy. So that's like the battery charge. And so when a cell needs to use energy for almost anything that cells do, they'll use the energy in that bond. So they remove the phosphate, a tiny bit of energy is released, and that's used by the cell for whatever it's doing that requires energy. And then to recharge that battery, the phosphate is put back on. So when there's only 2 phosphates, it's called ADP, adenosine diphosphate. Di means 2. So adding that phosphate back on is called phosphorylation. So how does that happen? How does a cell recharge that battery or put that phosphate back on? Or what does it use for energy to recharge these batteries? And that process is called aerobic cellular respiration, or ACR. Aerobic, anybody know what that means? Aerobic means it requires what? Oxygen. Right? If you do aerobic exercise, that's exercises that make you breathe fast and deep to take in more oxygen. And cellular, because it happens in cells. And respiration, everyone knows respiration means breathing, right? But respiration, and the word respiration in biology, it means not only breathing, but using the oxygen that we take in by breathing. And this is where the oxygen is used. This is why you're inhaling oxygen right now, and why you will die if you stop breathing oxygen. Because if this process stops, you will die within minutes. So aerobic cellular respiration means breaking down nutrient molecules from the food we eat, like sugars, and amino acids from proteins we eat, breaking those molecules down using oxygen. This requires oxygen to release the energy that was holding those nutrient molecules together, and that energy is used to recharge ATP. So here is the overall chemical reaction for aerobic cellular respiration here. It's actually way more complicated than this, but this would be the nutrient molecule that's broken down, and we use glucose as an example, but this could be other kinds of nutrients. It could be amino acids from proteins we eat. It could be glucose or fatty acids from fats that we eat, or it could even be nucleotides from DNA that we eat. We all eat a lot of DNA, because we eat other organisms that have DNA just like we do. So this is not a huge molecule, but it's got 6 carbon atoms, 12 hydrogens, and 6 oxygen. So there's energy in the bonds holding these atoms together in this molecule. And here's the oxygen you're inhaling right now. This is required for this to happen. And so this arrow represents several steps. It's not just one chemical reaction, really. But then the products are water molecules. So we are making new water molecules in our cells all the time. Every time your cells break down a sugar molecule for energy, it's making new water molecules. We call that metabolic water. Water molecules made by this kind of metabolism. In fact, there are some desert animals, little desert mice that live in the deserts in Arizona, that can go their whole life without drinking water. Even the food they eat, dry seeds and stuff, doesn't have much water in it. This is all the water they need, the water that they make themselves in their cells. Carbon dioxide, another product. This is what you're exhaling right now. This is where the carbon dioxide comes from that you're exhaling. But this is the reason for all this, to get the energy that was holding this molecule together into ATP, recharged ATPs. And from one glucose molecule, enough energy is released to store it in about 30, to recharge about 30 something, 34 or 36 ATP, depending on what kind of cell it is. So each ATP just stores a tiny bit of energy, but it's important. It's the energy that keeps us alive. Okay, talked about all this stuff already. So we need to look at in a little bit of detail about how this happens, how this aerobic cellular respiration happens. So there's some chemistry in here, but try to keep it simple. So there's 4 steps in this process: glycolysis, preparatory reaction, citric acid cycle, also called the Krebs cycle, and the electron transport chain. Before we look at these, we need to mention these 2 molecules called NADH and FADH2. Just think of them as energy storage molecules, like rechargeable batteries, just like ATP. That's how they're used in this process. Some ATP is made in these first 3 steps, a little bit, but most of the ATP is used in this last step, the electron transport chain. And the energy for recharging the ATP by the electron transport chain, that energy is energy that is stored in NADH and FADH2 in these first 3 steps. So what's happening in these steps is taking that original glucose molecule, and breaking it apart little by little. It's not just broken apart all at once and all the energy comes out and it's used to recharge ATP. It's like first that glucose molecule is broken in half in glycolysis. So the energy released is used to recharge some ATP, but it also recharges some of this stuff. And then in these other reactions, a little bit of ATP is made, but most of the energy released for breaking down that glucose molecule farther and farther is used for recharging these. And then in this last step, the energy that was temporarily stored in these molecules is then put into ATP to recharge it. So look at each of these steps a little bit. The first one, glycolysis. Lysis, in a word, means to break apart, break something apart. In this case, what's being broken apart is a glucose molecule. So here's glucose. Remember, C6H12O6. So it's a 6-carbon molecule, and it's broken in half. Now, that ends up as 2 molecules. Each one is 3 carbons, and that's called pyruvate. But by breaking that molecule in half, enough energy is released to recharge 2 of these NADH molecules, one here and one here. And it also recharges 2 ATP. It uses 2 ATP here. So you can see ATP going to ADP. That means that the energy is used from here. Now this is a dead battery, but down here we're recharging, making 2 ATP here and 2 ATP here. So we used 2, but we make 4, so we're gaining 2 ATP. Just in this first step, we've already made 2 ATP molecules and recharged some of this stuff. So this could be considered a metabolic pathway because this first reaction made this pyruvate. Now in the next step, the pyruvate will be a reactant and be further broken down. So this goes actually the next 3 steps. So the preparatory reaction. Here's that pyruvate that was made in the first step. It's broken down farther, removing a carbon atom and 2 oxygens from that, what was left in this pyruvate. And that becomes the carbon dioxide. So this is part of the carbon dioxide that you're exhaling. It's made by just taking a carbon and some oxygens off of pyruvate, releasing it. And now what's left is a 2-carbon molecule called acetyl-CoA. And the energy to do that, the energy is released by removing these atoms from here is used to recharge some more NADH, which will be used later. That energy will be used at the end for making more ATP. So this is called the preparatory reaction, or the prep reaction, because it's preparing for this next step called the citric acid cycle or the Krebs cycle. And this is actually a very, very complex, complicated set of reactions. Every biology major has nightmares about the Krebs cycle because if you take biochemistry or cell biology, you have to learn, spend 3 or 4 weeks just on the Krebs cycle, learning all of that stuff, all the chemistry. But for us, we just need to know that this acetyl-CoA it is then further broken down. So this is only 2 carbon atoms, And so one of the carbon atoms is removed along with more oxygen to make more carbon dioxide. Um, so that's more carbon dioxide than we exhale. But then what remains is it continues on and it combines with more acetyl-CoA. And when this remaining one carbon molecule combines with acetyl-CoA, it makes citric acid. That's why it's called the citric acid cycle. But then this citric acid is then broken down, released carbon dioxide, and then so it's a continuous cycle. One carbon dioxide is removed, but then what's left combined with more of this, and then that's broken down, released more carbon dioxide is removed. But with every cycle, some energy is released. And that energy is used to recharge NADH, recharge some NADH2, and make a little bit more ATP. So we've already made some ATP, but we've recharged a lot of this stuff. NADH and FADH2. So there's a lot of energy stored in these that's used in the next and final step called the electron transport chain. So this is showing what a mitochondrion looks like, and this is kind of important to understand that it's like a long oval or like a sausage or something, and it has two membranes, an outer one and an inner one. You can see them both here. And in between the inner and outer membrane, there's a thin space called the intermembrane space. Inter means between. So the intermembrane space is between the inner membrane and the outer membrane. And so the glycolysis, the first step, it takes place out here in the cytoplasm, but the prep reaction and the citric acid cycle and the electron transport chain are in the mitochondria. So this electron transport chain, we'll see it happens here at this inner membrane, and we'll see what happens here. I think it's best just to look at this diagram. So this is the inner membrane. The outer membranes would be out here. Membranes of the mitochondria. So this here is the intermembrane space between the inner and the outer membrane. And these are molecules, special protein molecules in this membrane. Just like, remember, the plasma membrane has protein molecules embedded in it that do various things. But what these special protein molecules do is they use the energy in this NADH and FADH2 that was stored in there in the first three steps of aerobic cellular respiration. These protein molecules are like pumps, and they use the energy from NADH and FADH2 to pump H+ ions, positive hydrogen ions. They pump it from in here, in the inside part of the mitochondrion. They pump it through this inner membrane into the intermembrane space between the inner and outer. And it's called the electron transport chain because these arrows here represent high energy electrons from NADH and FADH2 that these high energy electrons it's the energy in those high energy electrons that do that are used for the pumping the H plus through the inner membrane. So this is building up a high concentration of H plus ions in this space between the two membranes. And then that concentration of H plus ions will flow back through the membrane, but they come through another protein molecule in the same membrane. But this is an enzyme called ATP synthase. Remember, ASE on the end of a word usually means it's an enzyme. And synthase is an enzyme that synthesizes something. To synthesize means to make a molecule. And the molecule that's being made is ATP. So this tells us what this enzyme does. It's an enzyme that makes ATP. And how it does that, it uses the energy from these H plus ions flowing through it to recharge ATP molecules. So think of this like a hydroelectric power plant, an electric generating power plant at a dam. Those power plants work by the water behind the dam is allowed to flow through a propeller, make the propeller spin. The propeller is connected to a generator that generates electricity. This is like that. This actually has something in it that spins in that enzyme molecule, and so here the energy from NADH and FADH2 is used to move H plus through the membrane, and then H plus is allowed to flow back through to generate ATP. Now, I don't know why nature could not have come up with a simpler way to do this. Why couldn't have more directly taken the energy right from glucose and put it into ATP instead of going through all these steps? But this is what we got. This is what happens. So so we've seen where the carbon dioxide comes from that we exhale. In those steps of aerobic cellular respiration, the carbon atoms and oxygen are just removed a little at a time, breaking down that glucose molecule, and those atoms become carbon dioxide that we exhale. But where is the oxygen used, and where does the water come from? That's here at the very end of the electron transport chain. So these electrons, these high energy electrons, they flow from one of these molecules to the other of the electron transport chain, and the energy is used for pumping H plus. When they get to the end, they're not high energy electrons anymore; they're just regular electrons. But what happens to them? Well, they combine with H plus ions. So these are positive hydrogen ions, which means they have lost an electron, right? So this electron combines with this ion, so now it's not an ion anymore, it's just hydrogen. And then that hydrogen combines with oxygen, there's the oxygen that we're inhaling. And hydrogen and oxygen make water. So that's where the new water molecules come from that are made by aerobic cellular respiration. So this is all at the very end of the last step, the electron transport chain of aerobic cellular respiration. And why this is important, why we need to have this oxygen here, is that if these electrons coming through the electron transport chain have nowhere to go when they get to the end of it, if there's no oxygen there to combine with this hydrogen and these electrons, then everything backs up and this stops. The electron transport chain will stop making ATP, and this step is where most of the ATP is made. Those first three steps just makes a little bit of ATP, maybe 4 ATP. in those first three steps. This step here makes like 30 or 32 from one glucose molecule. So without this, you're just not making enough ATP to stay alive. So this here is why we have to inhale oxygen. This is called chemiosmosis. That's where chemiosmosis. This process here of pumping H plus through and then having it come back through that enzyme is called chemiosmosis. Okay, so there again is our overall reaction. So we've seen how this is broken down, where the oxygen is used, where the water molecules are made, and where the carbon dioxide comes from. And like I mentioned at the beginning, this doesn't have to be glucose. Your cells can use amino acids from proteins we eat. It can be broken down for energy. Remember, a fat or triglyceride molecule is made up of glycerol and 3 fatty acids. So those can be used for breaking down for energy as well. But those different molecules, other than glucose, they can enter this. They can some of them can skip the first step or the second step. So some like if it's fatty acids from fats we eat, they don't have to be broken in half by glycolysis. They can just go right into the it says pyruvate oxidation. That means the same thing as the prep reaction. And oxidative phosphorylation, that means the same as the electron transport chain, just adding phosphates. Okay, so any questions about that? Aerobic cellular respiration. Complicated, but it all makes sense. Kind of have to just sit in a quiet room and stare at that for a while. So that's how all the cells of your body make energy or get energy out of nutrient molecules. If that stops happening just for a few minutes, if you stop breathing, ACR stops happening. You stop recharging ATP. You have enough ATP in your cells to only last a few minutes, and then they run out. Cells run out, and the first cells. Which cells of your body do you think are the first ones to run out of ATP and die? When you die, all the cells don't die immediately at the same time, but it's the brain cells that are among the first to go. Within 5 minutes, the brain cells die and they don't grow back. Brain injuries don't recover by your brain cells dividing to replace the ones that died. So this is what happens with a stroke. Blood stops going to the brain for some reason. So oxygen and nutrients not delivered to that part of the body. A heart attack, something blocking the blood flow to the heart muscle, not getting oxygen and nutrients. It's not like strangling someone to death is not like in the TV or in the movies. I see it all the time. Somebody's strangling somebody. In 10 seconds, he's dead. It takes a minute. You have to strangle somebody for 4 or 5 minutes for him to be dead. Okay, fermentation. What's this about? We all heard of this, but this is another way of breaking down nutrient molecules to get energy out of them. But the difference is this does not require oxygen. This is not aerobic, um, and only certain kinds of cells can do this. There are we know that yeast can do that. Yeast is used for making alcoholic beverages and bread, baking bread. Um, and some bacteria can do this, do types of fermentation. We'll mention them later, um, but some human cells can do this, and that's your muscle cells, the cells of your skeletal muscles that bend and extend and twist joints. Um, Without oxygen. So when you use your muscles, you exert your muscles so much um, that they run out of ATP because your breathing and your circulatory system can deliver only so much oxygen at a time to your muscles. So if you're working so hard, your muscles are running out of oxygen. They can't make ATP anymore. That they're getting tired. That's why you get tired. That's called muscle fatigue. And uh, but your muscle cells can do something that other cells of your body can't do, and that's this fermentation. And it's called lactic acid fermentation because it makes lactic acid as a waste product or byproduct of this. And so if you use your muscles so much they run out of oxygen, they can switch to fermentation and keep making ATP, but only for a while and not very much ATP because the problems with fermentation in your muscle cells is instead of 36 ATP from a glucose molecule, this makes only two ATP. That's better than nothing, but not much better. But it'll keep your muscle going a little bit longer after they're running out of oxygen. But another problem is the lactic acid. Um, the lactic acid then builds up in your muscles, and that's what makes your muscles burn when you use your muscles to exhaustion. Some trainers and coaches, when they say when you're training, you need to go for the burn. That means use your muscles until you feel them burning. That means you've exhausted your muscles. But when you get to that point, your muscles have used up all the oxygen. Fermentation has caused lactic acid to build up. Now you're done. Your muscles are just done. You can't do nothing now. You just need to rest. Let the lactic acid be carried away by your blood. Let oxygen come back into your muscles. Recharge some ATP. That's called oxygen debt. You've used up all the oxygen, and that debt has to be repaid by adding more oxygen back into the muscles, and it takes a while. What's I going to say? Oh, your muscles have a couple of other tricks, a couple of other ways of making ATP that your other cells don't have. Just because muscle cells use so much ATP. We know your red blood cells have a protein called, what's that protein that carries the oxygen? What's that called in red blood cells? Hemoglobin. So the oxygen, when it's in your blood, it hitchhikes on the hemoglobin in your red blood cells. Well, you have something similar stored in your muscles called myoglobin. So you have these myoglobin molecules in your muscles storing oxygen, just so you have a little bit extra, in addition to what's being delivered by your blood. And also there's another molecule stored in your muscles called creatine phosphate. It has phosphate in it, so that phosphate in creatine phosphate can be very easily taken off the creatine phosphate and put onto ADP to make ATP. So it's a quick, easy way to recharge some ATP in addition to the aerobic cellular respiration and fermentation. You can buy creatine phosphate they probably have it at Walmart or nutrition stores have it. It's a powder, it comes in a powder, and bodybuilders, weightlifters mix it with milk and drink it before they lift weights to give their muscles some more energy. Okay. Yeah, this lactic acid fermentation, this is the same process used by some kinds of bacteria that are used for making cheese, yogurt, soy sauce, sauerkraut. The flavor of sauerkraut is lactic acid from lactic acid fermentation. And some bacteria and also yeast do this kind of alcohol fermentation. And it's called that because ethyl alcohol or ethanol is a byproduct or waste product of this, along with carbon dioxide. So this is used for making beer and wine and alcohol. So along with the yeast that you put in that mix, you have to put a bunch of sugar in there because that's food for the yeast to break down by fermentation. And beer is carbonated. Where does carbonation come from? Just halfway through the fermentation, you put the beer in bottles with caps on them, and then that allows the carbon dioxide to build up and carbonate the beer. Same with champagne, it's carbonated. Wine and whiskey, we just don't seal it. Just let the carbon dioxide escape, so it's not carbonated. When we bake bread, we put yeast in there. What's the yeast do? It ferments and releases carbon dioxide and alcohol. The carbon dioxide bubbles. That's what makes the bread rise. And we don't get drunk from eating bread. That's because as the bread is baking, and the ethanol that's produced, it just immediately evaporates. You can smell when bread's baking. You can smell the ethanol coming off of it. Baking bread smells kind of like beer to me. Okay, and there are also a few kinds of one-celled organisms that can do yet another kind of process for breaking down nutrients, and that's called anaerobic cellular respiration. And so fermentation is also anaerobic, but this is different. There's only some bacteria and archaean. It says sulfate reducing. That means these weird little one-celled organisms, they use sulfur instead of oxygen. They use sulfur the way we use oxygen. And so it's a type of cellular respiration, but doesn't require oxygen. And these are weird critters that live in places like the hydrothermal vents at the bottom of the ocean, where boiling hot gases and come out of the Earth's crust, and hot springs in Yellowstone National Park. These weird little creatures thrive in environments like that, and they use hydrogen compounds coming out of these hot gases, like hydrogen sulfide instead of oxygen compounds. Okay, we should take a break and then start looking at the next chapter, chapter 5, about photosynthesis. It's just equally fascinating or boring. couple things I mentioned. Oh, you guys know about the open or walk-in tutoring for biology? You could get in some, okay. So that's several times during the week. In this room, there will be a biology instructor for any student from any class to come and get help, any kind of help you need. I'm here from Monday 3 to 5. I forget when Miss Curtis is here. So she was here on the 18th and 24th. What day would that have been? I don't know. But so you can, if you need help with this class, you don't have to come to my session. Any of the other biology instructors can help you. Exams here, if you want to pick yours up, you can. The grades are on the back. If you don't want it, they'll recycle them. Should be alphabetical. You guys had a lab practical Tuesday, right? That was fun, right? Lab practicals are always fun. I didn't look at the grades, so. And if you didn't do great on this exam, it's just one exam. You can still do better. Plenty of opportunity to do better. I'd be glad to help you if you think that tutoring or just if what I usually do if a student comes for help, if I have the next exam ready, I'll just sit with a student and I'll go through. I won't tell you what questions are on the exam, but I'll just tell you what you need to know from each question, or I'll just tell you make sure you know everything that's on the exam. Yeah.
For the next lecture exam we have, will it be comprehensive of continuously building? Or
No.
Just chapter?
No, just 3 through 7.
I have a question. On here, I circled the correct answer, but you still marked it on the same answer than I circled.
No, I never looked at it.
Okay.
Yeah, you're right. I'm sorry if you see something that looks like a mistake. I make mistakes when I make them here. Sorry. I like to use Scantron forms. Scantron machine makes mistakes too, but there's only one Scantron machine on this campus, and it's upstairs in the suite of nursing offices, and that door's locked most of the time, and those nursing instructors go home early every day, I guess, because there's never anybody in there. And so in a small class like this, I figure it's not even worth doing Scantrons. But even on the Scantrons, I go through and make sure that everyone that the Scantron marked wrong is actually wrong, because if you erase something, Scantron can pick that up. It'll think that's still there. Okay, well. So chapter 5, photosynthesis. So it provides most of the energy required by living things on Earth, because it's at the base of the food web, on land and in water. Not only plants, but algae are photosynthetic. Many algae are one-celled, tiny little plankton. They're tiny, but there's so many of them. The lakes and oceans are full of plankton, and photosynthetic. And also there's a type of bacteria called cyanobacteria, cyan is a color, right? A bluish green color. And in fact, they used to be called blue-green algae, but they're not algae, they're bacteria. So that was a bad descriptive name for them. But they're photosynthetic. And again, they're very tiny, but there's just so many of them in the lakes and oceans everywhere. And they're at the base of the food webs. Everything else eats them or eats something that eats them and on up through the food web. So converting solar energy, light energy, sunlight, along with carbon dioxide from the air, water from the soil, or from the surroundings if it's an aquatic plant or algae or bacterium, to make organic compounds, sugars, fats, proteins. Okay, so just some examples of photosynthetic organisms. You ever see a lake or something that looks green like that? Those are algae. That's an unhealthy ecosystem there. Not supposed to be that much of that. We'll talk about that. It's called eutrophication. The last 3 chapters, when we talk about ecology, and we'll talk about how and why that happens. Okay, terms autotroph and producer, they mean pretty much the same thing. An organism that can produce its own food. So this is photosynthetic organisms. So a photoautotroph is a photosynthetic organism. There are a few weird one-celled organisms that do something called they're called chemoautotrophs, and they do something like photosynthesis, but instead of using energy from light, they use energy from small inorganic molecules. And again, these are weird bacteria and archaeans, but mostly autotrophs producers, they're photosynthetic organisms. Heterotrophs, consumers, that just means something that is not an autotroph, so it has to get nutrients from other organisms. So we have to eat plants or animals or fungi. So we are heterotrophs and consumers. So that includes herbivores that eat plants, carnivores eat meat or other animals, and omnivores eat both, plants and animals. Most humans are omnivores. So we mentioned in the last chapter that energy passes up through a food web from starting with the producers, which are eaten by herbivores, which can then be eaten by carnivores and so on. So the energy passes up through the food web, but the energy is also used by each organism along the food web. So here's the overall chemical reaction for photosynthesis. Should look pretty familiar, except backwards, right? So aerobic cellular respiration, we started with this, a nutrient like glucose, and oxygen, and the products were carbon dioxide and water. And energy was released and stored in ATP, but here in photosynthesis, carbon dioxide from the air and water are along with energy from sun. So this uses energy instead of releasing energy. And then the whole purpose of this is to produce organic compounds like glucose. And the plants exhale oxygen, release oxygen. So just like aerobic cellular respiration, it's not as simple as just one simple chemical reaction. There's a bunch of steps represented by that arrow. And we'll look at those steps a little bit. So here's just a graphic of a tree, which is photosynthetic, taking carbon dioxide from the air, and water from the soil, which comes in through the roots, and it moves all the way up through the trunk of the tree, out the branches, to the leaves, where it's used for photosynthesis. Requires sunlight, oxygen is released, and sugars and other organic compounds are made. And those organic compounds become the tree, become the molecules that make up the tree. And also they are used by the tree for food. So a plant does aerobic cellular respiration much like we do, breaks down those sugars for energy, much like we do. But so plants can do photosynthesis and aerobic cellular respiration. We can only do aerobic cellular respiration. We can't do photosynthesis. So plants use oxygen and give off carbon dioxide when they do aerobic cellular respiration, but they release more oxygen than they use, and they use up more carbon dioxide than they release. Okay, so the organelles within plants and algae that in which the photosynthesis occurs are called chloroplasts. Those cyanobacteria, the photosynthetic bacteria, they do not have chloroplasts because they're prokaryotic. Prokaryotic cells don't have organelles like chloroplasts, but they have the same, pretty much the same process. It's just not contained in chloroplasts in the cyanobacteria. Let's look at to describe these other terms. Just look at a picture. So here's a leaf. The leaves are where a plant or where most of the photosynthesis occurs. A leaf is broad and thin. It has a lot of surface area to absorb sunlight. So that's where most of the photosynthesis occurs. But any part of the plant that's green can have photosynthesis going on. So this will be if we take a little section of this leaf and look at it sideways, look at the edge, cut through and look at the edge. We see, so here's the upper surface of the leaf, and here's the lower surface. We use the word epidermis for those cells. Same word we use for the outer layers of skin on animals like us. But this mesophil, meso means middle, so that's the in-between, between the upper and lower surface. That's where where most of the photosynthesis occurs, in those mesophil cells. And so these little green organelles are the chloroplasts, and the chloroplast is shaped kind of like the mitochondria, like a long oval. And inside of a chloroplast, there are thylakoids. Thylakoids are the green, they're thin, hollow discs or flat disc-shaped things, and a stack of them is called a granum. So a stack of thylakoids is called a granum. This space in the chloroplast surrounding the thylakoids is called the stroma. We see this word here, stomata, or stoma is singular, stomata is plural. So that's an opening in usually the underside of the leaf. It's just a hole that it can open and close, but it's for allowing carbon dioxide to come in for photosynthesis and oxygen to be released that's produced by photosynthesis. So this word stoma is similar to this word stroma that has an R in it, but they're different. Different meanings. Okay, so photosynthesis says in 2 stages. For aerobic cellular respiration, we saw 4 steps. This we're going to divide it into 2 steps: the light-dependent reactions and the Calvin cycle. So this one works. It happens first, and then the Calvin cycle after that. These are sometimes called the dark reactions, but that's not a good name for them because they can. I mean, sorry. This is the Calvin cycle is called the dark reactions sometimes because it does not require light, but it happens whether there's light or not. So it doesn't happen only in dark. But these have to have. This is where the light is used in the light reactions. Um, and again, this uses ATP and NADH. These 2 energy storage molecules are used in photosynthesis. And so what happens here, the summary of what happens is, in the light-dependent reactions, light energy is converted into chemical energy that is stored in ATP and NADH. So a bunch of ATP and NADH are made or recharged in the light reactions. Then, in the Calvin cycle, the ATP and NADH are used. Their energy is used to make organic compounds like glucose. Then these molecules go back to the light reaction to be recharged, and use in the Calvin cycle again, and then recharged. So this ATP and NADPH are recycled over and over. But we need to see a little bit how that happens. First of all, what is light? Because it's important to understand what light is. Light is electromagnetic radiation, and that means electromagnetic radiation includes the radio waves, infrared, visible light, ultraviolet, X-rays, gamma rays. They're all the same thing. Electromagnetic radiation. It just means waves of energy moving through space, and it's in waves because as it's moving, the amount of electricity and magnetism increases and decreases. But you see that in these different kinds of electromagnetic radiation, the waves are longer or shorter. And so as these waves are moving through space, if they are far apart like this, there's going to be less waves hitting me per second, right? If they're short like this, there'll be a whole lot of them hitting me real fast per second as they're moving through space. So that's called the wavelength. It's how far it is from the peak of this wave to the peak of this one. And these are kilometers, meters, centimeters, microns, and so on. So the wavelength gets shorter. And the wavelengths mean really the same thing as frequency in here, because if there's a long wavelength, that means that the low frequency, the waves are not hitting me as many per second. But here, shorter wavelength means higher frequency. More wavelengths are passing a certain place in space per second. So these are low energy because fewer waves per second. This is high energy, more waves per second. So radio waves, these are the kind of electromagnetic radiation that is used by broadcasting radio. This is broadcasted by a transmitter. Your radio can receive that. If you have an old style radio that actually uses radio waves. Infrared, well, this here is visible light. The visible light, that's what we can see with our eyes, and just a little bit lower energy than that is infrared. And a little bit higher energy than that is ultraviolet, and those things come from the fact that the wavelengths of visible light are the different colors that we can see. And so the lowest energy, or longest wavelength, highest frequency is red, then orange, yellow, green, blue, and so on. So getting longer or shorter wavelength, higher frequency, higher energy, going from right to left. And then, so a little bit below red is infrared, what infra means. A little bit above blue, violet is ultraviolet, where those names come from. And then higher energy than that is X-rays, and then gamma rays. These are high energy, high enough energy they can penetrate through your tissues, and gamma rays can be dangerous to our health. So these are just the wavelengths of light that we can see. There, and if you may have learned this if you took physics. Red, orange, yellow, green, blue, indigo, violet. Spells out Roy G. Biv, like the guy's name, Roy G. Biv. That's the mnemonic that's used to remember the colors of the visible light. It's called the spectrum or the range. These are the colors of the rainbow. If you ever see a rainbow, the colors will always be in that order. But so what we see as light, what we see as light is, or how our eyes work. There's different colors of light coming into our eyes, and I've read that as many as 50% of college graduates think that humans' eyes function by shooting rays out at the object you're looking at. Maybe because they read Superman comic books where he's got X-ray vision rays, looks like rays shooting out of his eyes. But your eyes work by light coming in through our eyes, and what we see, the images that your brain forms from the light coming into your eyes, are just the different colors, and the different dark or light shades of those colors. And so all of the light, all of these mixed together, if you have all of the different wavelengths of light mixed together coming into your eyes, we see that as white light. So that's what sunlight is. It's white. It's got all these, and solar radiation also has ultraviolet and infrared in it. But what we can see is as white light, it's just all the different red, orange, yellow, green, blue, indigo, violet mixed together. But if what we see as colors, like I'm colorblind, but that looks kind of light blue to me. Is that right? So what I see, why that looks blue to us is that white light, with all the different colors of light in it, it hits that surface. All of those wavelengths of light are absorbed by this, except for the blue. The blue is reflected off and into our eyes, so we see only blue. What if something looks black, like this tabletop? That's because all the wavelengths of light are absorbed by this surface. None of it is reflected off, so we don't see any color, just black. So what about green? Plants. Plants are green, so that means that what we're seeing as green, that's all of the white light is absorbed by that green leaf of that plant, except for the green. Green is reflected, and so we see the green. Everything else is absorbed and used by the plant for photosynthesis. So for photosynthesis, the plant uses red, orange, yellow, not green, but that's reflected off the plant, and blue, indigo, and violet are all absorbed in photosynthesis. Right. A couple kinds of this chlorophyll is the green stuff in plants. So this is what absorbs the light energy for photosynthesis. And there's two kinds, A and B, and they're both similar. They just reflect and absorb a little bit different range of wavelengths. And then there's another group of these pigments, so-called pigments or colors, that some plants have, carotenoids. So that it reflects yellow, red, and orange, so that they appear red, orange, or yellow to us. And so in the fall, when the leaves are turning color, they're not really turning color. It's just that the chlorophyll A and B just breaks down. They're less stable molecules than the carotenoids. So the green chlorophyll goes away, and then what's left is the carotenoids. So the carotenoids were there all the time. They were just covered up by the green. So we saw green. Then when the green disappears in the fall, we start seeing the yellow, red, and orange colors of the leaves. Right before the leaves, the leaves die and fall off. Okay. Okay. So how does this photosynthesis work? It's kind of complicated. So the best way, I think, is just skip to this one very complicated looking diagram, but again, it all makes sense. It looks complicated, but it makes sense. So PS stands for photosystem. So there's photosystem 2 and photosystem 1. And this seems backwards, that photosystem 1 happens first, and then photosystem 2 happens first, and then photosystem 1. And I'm not sure why they got named that way. I read that this one was discovered or figured out first, and then this one, but that still doesn't make sense that they would number them that way. But anyway, so you see light coming in. This is these are molecules of chlorophyll that absorbs light, and the atoms in the chlorophyll have electrons in them, like all atoms, and so these electrons are energized by the light energy. That means they get moving faster and faster. Like the electrons spin around atoms, like spin around the nucleus on atoms. They get going faster and faster. They get so energized that you jump off of those atoms, and then they are passed through electron transport chains, just like the one in aerobic cellular respiration. Electron transport chains. These are high energy electrons, and energized by light energy, and as they pass through here, they the energy in these electrons is used for, again, pumping H plus through a membrane, and so storing a high concentration of H plus in here, and just like in aerobic cellular respiration, there's ATP synthase enzyme molecules in this membrane. or that allow the H plus to flow through. That flow of H plus, the energy from that is used to recharge the ATP. So this is just like aerobic cellular respiration electron transport chain. High energy electrons use pump H plus through a membrane, and then it's allowed to flow back through an enzyme molecule to generate or recharge the ATP. But we're not done yet. That was photosystem 2. Photosystem 1, those high energy electrons, they continue, well, there are electrons in this chlorophyll that are also energized by light. And they get moving faster and faster, and they jump off, and their energy is used by this enzyme called NAD plus reductase to recharge NAD. So again, light energy absorbed by electrons, then those high energy electrons are used to recharge NAD. Remember I said the light reactions, their purpose is to recharge ATP and recharge NADPH. So that's what's happening here. Now these molecules have stored energy that was in the sunlight, and that'll be used in the next step, the Calvin cycle, to make the organic compounds like sugar. But we'll need to look at some other stuff here. Why don't the chlorophyll run out of electrons? Their electrons are being, on its atoms are being energized and jumping off. Why don't they run out of electrons? It's because water molecules are being split apart to oxygen and H plus. And H plus, that means it's a hydrogen ion that has lost an electron. That's the electron that is used to replace the electrons that are always jumping off the chlorophyll. So this is where the water is used in photosynthesis. So if there's no water, you can't do this. These chlorophylls would run out of electrons. And this is where the oxygen comes from that a plant releases as a byproduct of photosynthesis. From right here at the beginning of photosystem 2 in the light reactions. Why don't these chlorophyll molecules run out of electrons? Because the electrons from this electron transport chain from photosystem 2, they continue on to replace the electrons in photosystem 1 that are always jumping on. Okay, simple, right? I've read that several years ago, I read that there were scientists somewhere who are, they see this. What we're looking at here, this is electrons flowing. What is a flow? What is a flow of electrons? It's electricity, right? Electrical current. So some scientists see this and say, that's a flow of electrons. That's electricity, electrical current taking place in all these plants everywhere. Why can't we tap into that and get free electricity? So this is just like a natural solar energy generator. Just takes light and turns it into electricity. And so they're trying to figure out how can we tap into that, get that free electricity out of it. As far as I know, nobody's been able to make that practical, but it would be cool. Okay, so now we got all this NADPH and ATP with energy stored in it, and now the Calvin cycle. And again, like that citric acid cycle, it's a cycle that well, it, in this case, the cycle produces some sugars, but what's left is uh, what's left is used in the next circuit of the cycle. Um, so come back to that in a minute. But this Calvin cycle can be divided up into three parts: the carbon fixation. So this just means taking carbon dioxide out of the air. So carbon fixation. Um, and then reduction in chemistry. The word reduction means that a molecule is gaining gaining electrons or hydrogen atoms. It's called reduction. Oxidation is the opposite. Molecule losing electrons or hydrogen atoms. And if a molecule gains a hydrogen atom, a hydrogen atom is just one proton and one electron. So if a molecule gains a hydrogen atom, it's gaining an electron with a proton along with it. But in both cases, reduction is gaining electrons. Seems backwards. It seems like reduction would be losing something, but in chemistry, it means gaining. There's a reason for that, but I don't remember what it is. Why it's called that. Um, so this is where the energy stored in ATP and NADPH are used to make the glucose sugar. So, and that happens by um, uh, high energy electrons or hydrogen atoms from these molecules get transferred into the sugar molecule to make the sugar. And then regeneration, that's the continuation of the cycle after the sugar is made. So carbon dioxide from the air combines with the, the leftover molecules at the end of the last cycle of the carbon cycle. They combine together and make a molecule. So now we now say the carbon has been fixed. It's now being combined with something else to make a molecule that's now part of the Calvin cycle. And using energy from ATP and NADPH added to this, it produces sugar molecules. And that's the reduction part. Adding energy shows here, removing this hydrogen from the NADPH, and now that's added to this molecule to make sugar. So that's called reduction. And then, but there will be some leftover molecules that, and that's called RUBP, and that will be combined with some more energy from ATP to make a molecule which then combines with more carbon dioxide to continue the cycle. I'm sorry, it's a terrible explanation, but this molecule that's left from the last cycle combines with carbon dioxide. That's the carbon fixation. And then energy from ATP and then NADPH are used to convert that into sugar. But there's some leftover molecule that then combines with some more ATP to make a molecule that combines with more carbon dioxide. This Rubisco is an enzyme that's used for that carbon fixation, to take the carbon dioxide out of the air. So here's a diagram showing the whole photosynthesis process simplified. So here's the thylakoids inside the chloroplast. And so here's where the light reactions happen. This is green, it has chlorophyll that absorbs the light. And so here's where the light energy is converted into energy stored in ATP and then ADPH, which is then used in the Calvin cycle. Um, so um, So this is, what happens here is basically taking this little carbon dioxide molecule, adding energy and hydrogen from ATP and NADPH to make glucose, which is C6H12O6. That's carbon, hydrogen, and oxygen in it. Now the energy from these two molecules is being used up, so they're dead batteries. They go back to the light reaction to be recharged and recycled. Simple, right? I'm comparing the overall reactions for photosynthesis and aerobic cellular respiration. Again, they're the reverse of each other. And these, through photosynthesis, the plant can make glucose like this, but it can also make amino acids for proteins and make fats, and cellulose, which is what plant fiber is. That will become tissues of the plant. So we already talked about this. Animals, we take in oxygen and release carbon dioxide in aerobic cellular respiration. Plants do photosynthesis, in which they take in carbon dioxide and release oxygen, but they also use oxygen and release carbon dioxide. But they release more oxygen than they use, and they use more carbon dioxide than they release. So plants take carbon dioxide out of the air and add oxygen to the air. There, a long time ago, before there were any photosynthetic organisms, before there were even cyanobacteria, we didn't have all this nice oxygen in the air and in the water dissolved in the lakes and oceans like we have now. It wasn't, and so it would have been impossible for any aerobic organisms like us to live back then. It wasn't until these tiny little cyanobacteria started producing oxygen by photosynthesis. And even though they were tiny, there were enough of them that over millions and millions of years, they added enough oxygen to the water became saturated with oxygen, and the oxygen was leaving the water into the air, and eventually we got this nice atmosphere full of oxygen. Okay, any questions about this stuff? I guess sometimes the class go through 12 or 12:10? 12. I guess we better, because we're behind schedule, we better start chapter 6. Oh. Reproduction at the cellular level. Oh no, more cell biology? Bummer. Okay. You guys know who Edward Abbey was? Probably not. Um, Back in the 1960s, um, in the United States and the world, uh, there was what what is now referred to as the environmental revolution. It's when really for the first time, um, many people were getting concerned about the effects of human activities on the environment. What was all this pollution doing to the environment and to us? And what about in the population growth? The human population used to be growing much faster than it is now. It's still growing way fast, but the percent of growth each year used to be more. And people were worried about that. When are we going to run out of planet for all these people? And are we going to pollute ourselves out of existence? And so Edward Abbey was one of these environmental activists. He was very outspoken, wrote books about it. He he wrote a book, several well-known books at the time. One of them was, I think, called The Monkey Wrench Gang. I never read it, but it was about environmental terrorists who would. The environmentalists don't like logging because logging cuts down forests, and so these characters in this book that he wrote were these environmental terrorists who would go and sabotage the logging equipment for the loggers. And so they would sabotage the bulldozers and the chainsaws and that these loggers would use, and so they would disrupt their logging practices. He was a kind of scraggly, bearded, hippie-looking guy, and I think I've read that he was pretty eccentric, and I've read that he had himself when he died instead of a coffin, he had himself buried in his pickup truck. That he loved this old beat-up pickup truck. He had himself buried in it, which I wouldn't mind being buried in my truck. I love it. But anyway, okay. So this chapter is about how cells reproduce by dividing, cell division. Some terms here: genome. It's just sorry. All of the cell's genetic information. So almost every type of cell in your body has the same 23 pairs of chromosomes, and each chromosome is made up of segments called genes. The gene is just a section of a chromosome with information in it. So all the information in your chromosomes is called your genome. Each person has a genome. We could say that each species has its own genome because even though my genes might not be the same as yours, there's the same kinds of genes. I have genes for eye color, like you do. Genes for, genes for all the same kinds of features, even though my version of that gene might be not the same as yours. Most of the information in genes is for making particular kinds of protein molecules. Remember, a protein molecule is made up of a bunch of amino acids in long chains. The gene tells the cell which amino acids to put together in what order along that chain to make a particular protein molecule. That's just a comparing a eukaryotic cell and a prokaryotic cell. Prokaryotic cell has a nucleus and all this other stuff, endoplasmic reticulum and mitochondria and stuff. Prokaryotic cells don't just have. They have usually one chromosome that's like a loop, and it's not in a nucleus. So what cells divide when a cell reproduces. Whether it's a one-celled organism like a bacterial cell, it'll divide in half. Now there's two of them. Cells in your body, that's how we grow and how we heal, and how certain types of our cells in our body, they just don't live very long. And so we have to be always making more of them. And that's done by cells dividing, cell division. And so in this chapter we see in some detail how cells divide. So each of your cells have these 23 pairs of chromosomes, and these are very long molecules. It can be 2 or 3 inches long, one molecule. But they're all folded up, they're all tangled up inside the nucleus of your cells. And so when a cell is going to divide, half of those chromosomes go into one daughter cell, the other half go into the other daughter cell. If those long tangled chromosomes, they're moving into the daughter cells, that would be hard to pull them, for them to move into the daughter cells if they're all strung out and tangled up like that. So to make that happen more simply is before a cell divides, the chromosomes coil up, so they're short and thick. And so this is a DNA molecule. But here, when the cell's getting ready to divide, it'll coil up like a spring, or an old-style telephone receiver cord. And so it'll coil up like a spring, and then that spring will coil up again, so it becomes even shorter and thicker. So now, instead of a chromosome being like this, it's like this, short and thick, all coiled up. And why it looks like the letter X here is because another thing a cell does before it divides, it copies every chromosome. That way, because this cell has 23 pairs of chromosomes, then it divides each of these, what we call the daughter cells. If they're going to have 23 pairs of chromosomes also, then these chromosomes have to be copied. So that's why it looks like the letter X. There's actually 2 identical chromosomes stuck together. They're still stuck together. And now when the cell divides, these 2 chromosomes will separate and go into the 2 daughter cells. So all 46 chromosomes will be copied like this, and then they separate into the daughter cells when the cell divides. So that's what's happening here. The cell is coiling up, getting shorter and thicker, and also it has been copied and replicated, so there's 2 of them stuck together. So the rest of the time, when a cell is not dividing, the chromosomes don't look like that. So they're just long, strung out, real thin threads that you can't even really see them at all under a microscope. This histone proteins. These are protein molecules that are attached along the DNA molecule, and their function is they're the ones that's how the DNA coils up. These proteins, histone proteins, do make it coil up like that. But the rest of the time, those histone proteins aren't doing anything. So we're talking about human cells here. human cells have 23 pairs of chromosomes. Other species of animals and plants have different numbers. So a dog, I forget what it is, a dog might have 19 pairs. A horse might have 27 pairs. But for humans, it's 23 pairs. And um, so every type of human cell that is not a sperm or egg cell is called a somatic cell. And so when I say that human cells have 23 pairs of chromosomes, I'm talking about somatic cells because a sperm cell and an egg cell don't have 23 pairs of chromosomes. They have just one set of 23 chromosomes, not 23 pairs. And that's because when a sperm fertilizes an egg, now it's 23 plus 23, you're back to 23 pairs in that fertilized egg. So the sperm and egg are the only kind of human cells that have just one set of 23. All the others have 23 pairs. This word karyotype just means this. It's just if somebody in a lab took all these randomly arranged chromosomes out of a human cell and arranged them like this, the cells are or the chromosomes are numbered from longest to shortest, and there's a pair of each. So you got a pair of number 1 chromosome, a pair of number 2, and so on. But when they're arranged like this, this is called a karyotype. Just all the chromosomes in a human cell arranged like this. So the two chromosomes of a pair are called homologous chromosomes, and they all have the same kinds of genes on both chromosomes of a pair. So, for example, if right here on this number 1 chromosome there's an eye-colored gene, then there's an eye-colored gene in exactly the same place on the other number 1 chromosome. That's called the locus. The location on the chromosome is called the locus of a chromosome. So they both have eye-colored genes in the same place, but they're not necessarily the same eye-colored gene. One could be for blue eyes, and the other could be for brown eyes, or they could both be for blue or both be for brown. What else? Yeah, autosomes and sex chromosomes. So these all have numbers, but this one just says X. So these are the sex chromosomes. The others are called autosomes or autosomal chromosomes. But these are called sex chromosomes because these are the ones that have information about information for producing the reproductive organs. These are both X chromosomes. Look at this other karyotype on the next slide. This one has an X and a Y. The 2 X chromosomes are the same size. One's 10, but the other's 3. But they're the same size. They're homologous, just like the others. They have the same kinds of genes on them. But comparing the X and Y, Y is much smaller. They're a pair, but they're not really homologous, but they don't have the same kind of genes on them. So this would be a male, because it has an X and a Y. This is a female, 2 X's, or XX. And so the X chromosome is much larger than the Y chromosome, but that's because this has on it the information for how to develop the female reproductive organs. So, but I also have an X chromosome. I don't have female reproductive organs, but whether you're male or female, you have an X chromosome. So when you are a little embryo and you start developing reproductive organs, they'll start developing the same, whether you're male or female. They look the same. But at some point during the pregnancy, the information on the Y chromosome, if there is a Y chromosome, that information will take over and direct the development of those reproductive organs to become male reproductive organs instead of female. If there's just two X's, they'll continue to develop as female. There's other stuff, other kind of gene on X chromosome. It's not only reproductive organ information. When we do genetics chapter, we'll see that. What else we need to talk about? Oh yeah, this word alleles. It just means different versions of the same kind of genes. Like I was saying, you could have a blue I allele on one chromosome and a brown I allele on the other one. We would say those are alleles, for the different alleles for the eye color gene. So there's a blue eye color allele and a brown eye color allele, but they are both eye color genes. And because these are short and thick, we know that this karyotype was made when that cell was dividing, because otherwise these long threads, they wouldn't look short and thick like that. This word sister chromatids here, again, this cell's getting ready to divide, so it's got the two identical chromosomes still attached to each other. Plus, every chromosome has been copied. They're still stuck together, and when they're still stuck together, these are called sister chromatids. They're just the identical chromosomes. And where they're stuck together is how it's centered here. Okay, cell cycle. This is just the what happens during the life of a cell. You have a life cycle. You're born, and then you're a child for a while. You grow, and then you become an adult, and then eventually you may reproduce and make more humans, and then you die. Cell cycle is just what happens from the time the cell is formed during its life, and then when it reproduces, and then it dies. So that is represented by this pie chart here. And cell cycle can be divided into two parts. Interphase, so this is all interphase, all except for this little part here, called the M phase or mitotic phase. Mitotic means cell division. So this is when the cell is dividing, and this is the rest of the stuff. So the life of a cell begins when it is formed by cell division. So here's the parent cell, and then these are daughter cells after cell division. So this is the beginning of this cell's life. And then it'll live on for a while, and eventually it will divide. And then that will be the end of this cell's life, the beginning of these two daughter cells' life. Or this cell will just die. Some cells never divide, they just die eventually. But what happens during the life of this cell is called the cell cycle. So here's a cell division, so the beginning of a cell's life here. And see this cell divided, and now the daughter cells will be half the size of that parent cell, right? And so that new cell will have to grow to get to be normal cell size. And that's what happens in this G1 phase. G stands for growth. So after a new cell is formed by cell division, it has to grow. So that's the G1 phase. It'll have to grow and make more organelles to get started in its life. And so it can live for a while, and then it'll be time for it to divide. And so before it divides, so it has to copy all of its chromosomes, right? So because this kind of mitotic cell division we're talking about, the daughter cells are identical to the parent cells. They'll have the same number of chromosomes. So DNA synthesis. S is for synthesis. Synthesis means to make molecules, and what's being made is chromosomes, a copy of every chromosome, DNA synthesis. So now the cell is getting ready to divide, and before it divides, it'll have another growth phase called G2. And so the cell will grow a little bit and maybe make some more organelles so that when it divides, the two daughter cells will have enough organelles to get started in their lives. Then it'll go into the M phase, where the cell divides, and it says mitosis and cytokinesis. We'll look at those processes in a little more detail later. But mitosis is actually means a division of the nucleus, the chromosomes separating. And cytokinesis means division of the rest of the cell, everything else, the cytoplasm and the plasma membrane. But that would be the end of the life of the cell in divides. But this G0 has an arrow going off to the side. That represents like after the G1 phase. Now the cell is full grown, and if it just goes on living in your body, for example, as a cell in your kidney or something. It just lives on there for years. That's what's meant by the G0 phase. It's not growing, and it's not getting ready to divide. It's just living its life as a cell in your body in that G0 phase. So it could do this for a long time, years and years. Maybe something would trigger it to divide, like there's an injury or something. Now it has to divide to replace some cells that were killed off in an injury. Then they would go from G0 into S phase. Okay, we talked about the chromosomes are copied or replicated or duplicated before the cell divides during S phase. So looks like this. Looks like the letter S because there's two identical chromosomes still stuck together, and then when they separate during cell division, now they're not called sister chromatids anymore. Each one is an independent chromosome. Okay, so what happens in this M phase, the mitotic cell division? What we're talking about in this chapter is mitotic cell division. There's another kind of cell division that happens in eukaryotic cells, like human cells, called meiotic cell division. So what we're looking at is mitotic. The other kind is meiotic. Similar words, but different. So mitotic cell division is just a cell dividing to make more cells. This is what happens for growth, for growing, and for healing, and for replacing cells that just wear out and die. And this happens in almost all the cells of your body, except for making sperm and egg in the testes and in males and in the ovaries in females. So this is mitotic cell division, but for making sperm and egg is meiotic cell division. And it's a little different because in the case of mitotic, the daughter cells are just like the parent cell. But in the case of meiotic cell division, it starts out with a cell with 23 pairs of chromosomes, but then we end up with sperm and egg, which have only one set of chromosomes. So how does that happen? It's because there's two divisions, two sets of divisions. This original cell in the testes or ovaries divides, makes two daughter cells, and then there's a second division. And the chromosomes were duplicated only one time. So, but there's two divisions, so we end up with, started out with 23 pairs of chromosomes. Now there's only 23 chromosomes in each of these. If this was in a male testes, each one of these daughter cells would grow a tail to become a sperm cell. So this is meiotic cell division, two divisions. It's just in females, it's a little different. Same kind of thing, two divisions, but in the ovaries of a female, after the first division, one of these dies. And then the remaining one, the surviving one, divides the second time. And, one of these dies. So there's only one egg that results from meiotic cell division in females. And these ones that die are called polar bodies. This is the first polar body, this is the second polar body. So the next chapter we'll learn about meiotic cell division. But what we're talking about now here is a mitotic that happens in all the other cells of our bodies. Okay, so there was a mention in the cell chapter about centrosomes. These are part of the cytoskeleton that they only function when a cell is dividing. And they make these protein filaments that attach to the chromosomes and move them into the daughter cells. And that's called the spindle apparatus. These protein filaments that move the chromosomes around during cell division. That's the only time that these centrosomes function. The rest of the time, they don't do nothing. So here's a summary of mitotic cell division. And it's shown only two pairs of chromosomes to make it simple. If this was a human cell, there would be 23 pairs of chromosomes. And it's showing each pair, blue and a red one. So we can say the long ones are the number 1 chromosomes, and the short ones are the number 2 chromosomes, and there's 2 of each. So the cell's getting ready to divide, so first thing it's going to do is copy all of its chromosomes. Now there's 2. Each of those chromosomes has been copied, so there's now 2 sister chromatids, or 2 identical chromosomes, still stuck together. And then when the cell divides, that's when the sister chromatids separate into the daughter cells. Now, each daughter cell is just like the parent cell. There's 2 sets of two pairs of chromosomes. That's what this N, the letter N here, just means the number of chromosomes in one set. In this example, N equals 2, because there are only 2 kinds of chromosomes, number 1 and number 2. If this was a human cell, N would equal 23. So there would be 2 sets of 23, or a total of 46 chromosomes in a human somatic cell. This is called duplication. Mitotic cell division is called duplication division because the daughter cells are just like the parent cell. Okay, practice questions. The phases of mitosis. The mitosis can be divided up into these there's five stages listed there, and so some books list only 4 phases. The book we're using now includes an extra pro-metaphase. I don't know why they do that. When I was in high school and college, and it was always prophase, metaphase, anaphase, and telophase. But some books have to be different and add an extra phase just to confuse people, I guess. Did you guys learn it in high school? There were 4? Yeah. So, so most books combine these two as just prophase. So this is just in more detail what happens going from this to this in that division. So prophase. The chromosomes can condense and become visible. Condense means they coil up and become short and thick. And so you can see them under a microscope, like this, with like the letter X. These spindle fibers emerge from these centrosomes. So these are the centrosomes here, and these lines coming out of them, those are those protein filaments that are extending out, and they're going to grab onto the chromosomes. The nuclear envelope breaks down, so the membranes surrounding the chromosomes disappear, because the chromosomes would not be able to separate into the daughter cells if they were inside of a nucleus like that. The centrosomes move toward opposite poles, so these centrosomes will end up opposite each other, like here. So in what they call pro-metaphase, which is really just later on in metaphase, these kinetochores, that's just where the protein fibers attach. You don't need to know that word. Next phase, these spindle fibers that are attached to the chromosomes now, they move the chromosomes around until they're lined up right in the middle of the cell. This is called metaphase. Just remember, middle. The chromosomes are in the middle of the cell during metaphase. That's called a metaphase plate, right? This just is an imaginary plate in between the 2 ends of the cell where all the chromosomes will line up. So if you look at a cell under a microscope and you see all the chromosomes in a dark line right down the middle, you know that cell is dividing and it's in metaphase. Next one is anaphase. The 2 chromatids are being pulled apart, so they start to separate. As you can see, from a little bit of daylight in between 2 lines of chromosomes. That means it's in anaphase. And then later on in telophase, the chromosomes are completely separated into 2 sets, and a new nuclear envelope will form around each set of chromosomes, forming two nuclei now. And spindle fiber breaks down, it's not needed anymore. And meanwhile, during telophase, the rest of the cell changes in half, and that's called cytokinesis. And now there's 2 separate daughter cells. And that's in that pitching in half is called a cleavage furrow, a groove. In plants, that doesn't happen. Instead, in plants, instead of the cell pitching in half, there's the cell plate forms, It's just a new layer of plasma membrane and cell walls formed in between the two daughter cells, and that separates the parent cell from the two daughter cells. Okay, that's enough. Bye. Haley, I'm gonna just change your grade. I'll go into Moodle right now and fix it, so you can keep this.
Thank you.
Good grade.
That's good, we can keep it.
Bye.